Hello friends, today we will discuss about nanoparticles in drug delivery. Myself Dr. Vinay Singh, Department of Pharmacy, SGSITS Indore. My colleagues are Nitish Gupta and Mr. Suresh Kumar Paswan and Prakash Kumar Soni. All we have contributed to this session of nanoparticle in drug delivery. Nanotechnology and nanoscience have great potential in area of research and application. With the development of nanotechnology, it is now possible to use the nanoparticle in area of drug delivery. Today we will discuss nanoparticles in drug delivery. Talking about the session of nanoparticles and fundamental of nanoparticles, we will discuss or we will cover these topics: nanoparticle size, nanoparticle surface, nanoparticle suspension and settling ability, and the optical properties of nanoparticles. Let us see some figures related to the sizes. The smallest object which can be seen by eyes is about 100 micron. The thickness of human hair is 50 micron. You can see here, and the size is 50 micron. The size of RBC is 5 micron. Size of bacteria is 1 micron or 1000 nanometer. Size of virus is 100 nanometer. The ribosome which is present inside the cell is 10 nanometer. The DNA is 3 nanometer and the size of carbon atom is 0.1 nanometer. Now let us discuss about nanoparticle surface. The percent surface molecule in the particle is the key and fundamental property while discussing the nanoparticle surface. The number of molecules present on the particle surface increases as the particle size decreases. It is interesting to see that for a 10,000 nanometer or 10 micron, that is this, a very small percentage of molecule, that is 0.03, are present on the surface, and hence the drug dissolution rate is much lower for microparticle when compared to the nanoparticle. You can see these figures. The nanoparticle as compared to microparticles shows a strong adhesion due to increased percent surface area. Now let's discuss about nanoparticle suspension and settling ability. Particle settling velocity should be the key factor while discussion of the settling ability. The settling of particle is governed by Stokes' law, where settling is directly proportional to the size of particle. The higher the size of particle, the higher will be the settling velocity. You can see by these examples that as we go higher in the particle size, the settling velocity will go higher. Now let's discuss about Brownian motion of the particle. According to the Einstein's fluctuation dissipation theory, the Brownian motion is inversely proportional to the size of particle. You can see the Brownian motion of 1 micron particle, that is 1000 nanometer, it is 1716 millimeter per second, 1 nanometer per second. The Brownian motion is high as compared to the settling velocity. You can see that this is again the 1 micron particle and the settling velocity is 430 nanometer per second. Hence the particle lesser than 1 micron, they have good suspension ability. Now let's discuss about the optical properties of nanoparticle. The topic is also size dependent color variation of gold nanoparticle. Gold and silver nanoparticles show size dependent of optical property. The intrinsic color or nanoparticle changes with the size because of surface plasmon resonance. Such nanoparticles are useful for molecular sensing, diagnostic and imaging application. For example, gold nanoparticle can exhibit different colors based on sizes. You can see the comparison between nanoparticle size and their absorption maxima. of gold nanoparticle solution in water. They have varied absorption maximas. Now, let's discuss about the production of nanoparticle. It is challenging to produce nanoparticle of materials of soft or hard in nature. The soft materials are sticky in nature. The powder available in bulk have large size, which are in millimeter, and some of them can easily solubilize to molecular level. Hence, there are two extremes, which is in molecular size and millimeter size. Hence, to obtain nanoparticle in the 50 to 300 nanometer range for drug delivery, one requires of the order of 104 to 108 molecules in each particle. The size has to be achieved from either solution phase, which is single molecule, or millimeter size particle having 10,000 molecules. The first approach, where the particle is broken down to nano size, and whereas the second approach, the particle will be built up from the molecules. The two general approaches for the production of drug nanoparticle are very well depicted in the figure. The detailed study of process we will study in other lectures. Now, let's discuss about the characterization of nanoparticles. Basically, there are two types of characterization. One is size and other is zeta potential. While dealing with measurement, there are basically two methods: dynamic light scattering and electron microscopy. In dynamic light scattering method, the measurement is done on the basis of Brownian motion. The diffusion coefficient is calculated on the basis of a scattering and autocorrelation. You can see that, or you can correlate that in this curve there are broad peaks, and they can be correlated to the large particles, while this one is having sharp peaks, which can be correlated to small particles. The particle size is calculated from diffusion coefficient from Stokes-Einstein equation, that is D is equal to KT divided by 6 pi meter R. Another method, an interesting one, is electron microscopy. The scanning electron microscope is a microscope that uses electron instead of light to form an image. It has many advantages over traditional microscope. The SAM has a large depth of field, which allows more of a specimen to be focused at one time. The SAM also has much higher resolution, so closely spaced specimens can be magnified at much higher levels. Here the electromagnets are used instead of lenses. It has higher control in the degree of magnification. The picture obtained here are crystal clear. Now, let's discuss about zeta potential. One of the interesting property of nanoparticle, which defines its stability. The zeta potential is the electrical potential in the interfacial double layer at the location of the slipping plane relative to a point in the bulky fluid away from the interface. The zeta potential is a key indicator of the stability of colloidal dispersion. The magnitude of zeta potential indicate the degree of electrostatic repulsion between adjacent similar charged particles in a dispersion. For molecules and particles that are small enough, a high zeta potential will confer stability. Here is one of the most conventional method used for measuring zeta potential. You can see that a particle travel between two electrodes, and it can be seen through a microscope, and accordingly the migration of particle is correlated with its zeta potential. That is, the conventional method for measuring zeta potential is to observe the migration of particle in a dispersion with a microscope and measure their velocity using a stopwatch. The current technique of laser Doppler velocimeter has mostly replaced this laborious measurement procedure. Now, let's discuss some of the application of drug nanoparticles. Nanoparticles are chiefly used in chemotherapy, vaccine delivery, protein and peptide delivery. Nanoparticles also have impact on ocular delivery and brain delivery, also very important in DNA delivery. Nanoparticle in chemotherapy can be very well depicted from this video. That is ambroxan, which is an injection suspension of nanoparticle.
A new paradigm for the treatment of cancer has been developed that leverages the tumor's feeding mechanisms against itself. This paradigm is known as albumin-bound nanoparticles. Each nanoparticle is about 100 times smaller than a red blood cell and is composed of an outer layer of albumin surrounding an inner core of the water-insoluble cytotoxic agent. Due to their small size, these nanoparticles, trillions of them, can be safely administered intravenously and rapidly in only 30 minutes. Once in the circulation, albumin-bound cytotoxic agent bind GP60 receptors on vascular endothelial cells in exactly the same way that normal albumin does. The activated GP60 receptor interacts with caveolin-1 protein, leading to formation of caveolae. The caveolae then transport their cargo, albumin loaded with cytotoxic agent, to the tumor's interstitium, where it is trapped. For the cytotoxic drug to be...
You have seen that how efficiently a nanoparticle has contributed to the drug delivery. Now, another example of or application of nanoparticle is in vaccine delivery. The oral vaccines suffer the exposure to the harsh environment of stomach and intestine and gets degraded. These can be encapsulated in the polymeric nanocarrier, which save them from degradation, and absorbed from the pairs patches. You can see pair patches in the intestine, present in the intestine. These pair patches are higher in number in children. Another important application is in protein and peptide delivery. The encapsulation of nanoparticles protect these protein polypeptide against proteolytic enzyme. One of the most important application of nanoparticle is in ocular delivery. The conventional eye drops drains out easily. The drug-loaded nanoparticle with high payload adheres to the inflamed tissue and increase elimination half-life and prolong and maintain intraocular pressure. Another important application is in brain disease. The drug, including antibiotic, anti-neoplastic agents, and neuroleptic agent, have hurdles in crossing blood-brain barrier. This can be overcome by encapsulating these drugs in polysorbate coated nanoparticles. Another important application of nanoparticle is in DNA delivery. The nanoparticle improves the stability of DNA in bioenvironment. These are the references from we have taken these material. Thank you.